welcome everyone to another Solanaceae seminar online this Friday. And today we are very happy to welcome Andrés for his talk, who is also a co-organizer of these seminars. So Andrés has been working in Solanaceae almost since he was born. He did his undergraduate degree at the National University of Colombia, working on spiny Solanum, and then he continued a master's also in the National University of Colombia. And during his master's, he was a research visitor at Lynn Bo's lab in Utah, working in the phylogenetics of Solandreae. And then after his master's, Andrés moved to Edinburgh in order to do his PhD at the University of Edinburgh, working with Gino Sarcophisa and Sandinab. He has been focused on genomics, phylogenetics, morphological evolution, and taxonomy of the amazing tribe Solandreae. Last year, he moved to the Jardín Botánico de Bogotá, where he is the current curator of the plant collections. And today, he's going to talk about his doctoral thesis on the epiphytic species of the Nightshade family, and his talk is entitled Roots in the Clouds, Evolution of Epiphytes in Solanaceae. So thank you, Andrés, and you can start sharing your screen.
Thank you, Rocio and Chelsea, for this invitation. I'm so glad to be here today to present my PhD work in the tribe Solandreae. I'm going to talk for, I don't know, 40 minutes, maybe 50, something like that. And I first at all, I want to say thank you to the Royal Botanic Garden of Edinburgh and the University of Edinburgh, and also to the Jardín Botánico de Bogotá for all the support. And okay, we're gonna start. First, I want to talk about epiphytes in general, and the concept of the epiphytes come from Schimper from 1888, and the definition that is still in use. This is an epiphyte is a plant that germinates and root non-parasitically on other plants at all stage of life. And this last part, and all stage of life, is very important, and it's good to have this to take this in account, because we can have some variation of this life form within epiphytes, like the epiphytes that are plants that germinate on the canopy on the top of the trees, and secondarily they produce roots that extend to the ground and enter in contact with the ground. We also have nomadic vines. That is quite a new concept, more or less like last 20 years that people are using this concept. And this is this plant that they start to grow from the ground and start to climb to the trunk of the trees, and secondarily lose the contact with the ground. And we also have facultative epiphyte that are plants that can survive growing on the ground, or can survive living like an epiphyte. We're gonna talk a little more about this later. And I also had here two illustrations from the original work from Schimper. He visited Venezuela. He visited so many places in the world, and he saw some bromeliaceae growing in South America. And this one was the illustration that he used to illustrate his concept of the epiphytic plants. Okay, this group is quite important. Is represent the 9% of the all vascular plants in the world, and more or less 28,000 species in so many genera and 73 families. But almost 90% of the epiphytes are angiosperm and 10% ferns, and we only had one gymnosperm, that is Samia pseudoparasitica, that grow in Panama. It's endemic from Panama. But this epiphytic life form is not occurring in the same amount of species in every family. Is completely unbalanced in 4 families. We had the 80% of the epiphyte occurring in only 4 families. Of course, the Orchidaceae is the family with more epiphytes. I think from this 2000, this 28,000, like 18,000 are Orchidaceae. We also have a good amount of epiphytes in Piperaceae, in Bromeliaceae, and also in the family Polypodiaceae. Okay. The distribution of the epiphyte follow more or less the distribution of the tropical forest. I'm gonna talk later about why the epiphyte needs or are more they are highly diverse in the tropical forest. And I put here just two example of this distribution, the epiphytic cacti that are mainly tropical. I have some species in Africa, Madagascar, following the tropical forest, and the same happened with Drinaria. This is a genus of Polypodiaceae that is more centered in the old world, but also in the tropics of the old world. Okay, these plants to survive like an epiphyte growing without contact to the ground had to they had to be efficiently taking nutrients because they don't have access to the ground, to the soil, to can capture the nutrients. Because that the epiphyte developed a lot of adaptations to deal with the epiphytic life form. One of these is the photosynthesis that is occurring in several families. For example, in cactus, in so many families of epiphytic plants. But also, for example, some family had very specialized adaptation, like Bromeliaceae that had this kind of foliar trichomes. Orchidaceae had these filamentous roots. Then I think some ferns also had some kind of these roots that works like a sponge to capture the water from the mist, from the rain. A lot of the species have this kind of thickening of the stem or from the roots. That is, this case is a limnotuber, but could be also a root tuber or stem tubers or thickener rhizomes to storage nutrients or to storage water for the dry season, for example. Bromeliads also have this kind of adaptation, this leaf arrangement to can capture the water from the rain and also some nutrients that are dissolved in this water. Orchidaceae have pseudobulbs, and for example, this is a Piperaceae that has succulents. Succulents is also very common in several families of epiphytes. To capture these nutrients, these species had they use, for example, this debris that is in the tree in the branches of the trees. For example, here is a lot of material there that this is kind of aerial soil, and they can capture nutrients from there. Also can capture nutrients from the atmospheric inputs like wet deposition with the rain or dry deposition from the dust, for example. Some bromeliads can do that. And also some epiphytes had association with ants to can supply the nitrogen or the nutrient that this plant need. Another problem that had the epiphytic plant is the light exposure. Is one of these constraints. To deal with that, these plants had some adaptations too, like photosynthetic non-foliar organs. For example, this orchid is a leafless orchid from, I think it's from Malaysia, and the roots are photosynthetic. The same happened with the stem in so many cacti. For example, this is a Solanaceae that had cluster leaves to increase the exposition of the leaves to the light. And you can see here, this is just a representation of typical forest, and some epiphytes are in the top, in the canopy, in the top of the trees, but others grow in the understory forest. According to this, these plants can develop different adaptation to can capture light to do photosynthesis. Also, the reproduction is quite challenging in the epiphytic life. So many epiphytes grow in a very small population, very scattered across, along in the forest. For example, I had here these two representation from two. This, imagine that this is 3 kilometers from here to here, and this species had to reproduce, had to transfer pollen from this plant to the other plant, and this is a huge challenge for some epiphytes. Um, sorry. So many epiphytes had a solution for this problem. For example, the trapline pollination with, I'm going to explain this in the next slide, but so many families develop this specialization with pollinators to come pollinate the flowers. Also, deception pollination in orchidaceae, that is very common. And some plants that can, for example, some plants that don't have access to pollinators can self-pollinate. That is quite, it is not very common, but it's happening in some orchidaceae and in some bromeliaceae. And we can have also mid-set system where some plants can self-pollinate, but also can use some pollinators to transfer the pollen. This is the trapline pollination. That is quite one of the commonest, I think is the commonest pollination, like a specialization in epiphytes, where we had this kind of, for example, bees that bees have memory and can remember the path that they follow every day to visit the same flower, the same with the hummingbirds and with rodents or with these moths or bats. This is a renewable. Okay, this is a resort that they can exploit every day, and for this reason, this is association is quite important in epiphytic plants. Okay, and talking about this kind of trapline pollination, the pollination syndrome is a good concept I have to introduce now because I want to talk later about that. The pollination syndrome is an integrated suite of reproductive characters that match flowers to functional pollinator groups. And I put here example with the bat pollinate, with the syndrome of pollinated by bats. The functional group is bats. We can see here, so convergent, this is a passiflora with a Solanaceae, with a Dibindoniaceae, I think this is an Acanthaceae, Polemoniaceae, this is oh, I forgot the, no, Centropogo, no, Burmestera. And each functional group, in this case, bats, exert different selective pressure on this species of plants to convergence to the same functional pollinator, or maybe to diverse to different. Close related species could be very different in the flower morphology because they have different functional groups of pollinators. This is a concept that it could be useful in some groups, but also it is a lot of criticism with this concept because some species could be pollinated for more than one functional group. And for it is a lot of literature about criticisms, but also some publications support this concept. Okay, now I'm gonna enter to the Solanaceae family, finally. The Solanaceae is a quite medium-sized family distributed globally with 100 genera and 2,700 species. And the Solanaceae are mainly shrubs, sometimes herbs. I think the herbs are very common. A few trees, vines, and lianas are quite common too, but rarely they are epiphytes. And we are going to talk today about the epiphytes. This is just an example of the life forms in Solanaceae. We can have this kind of these huge trees. For example, this is a 30-meter tree, Solanum sycophantha, that's growing in Colombia. This is a crepy vine that is Solanum ebulifolium that climb the trunk of the trees. This is an example of an epiphytic Solanaceae. This is a different family of tree, but the Solanaceae is this here that is attached to the trunk here. This is a big liana, also in Solanaceae, and we have shoots. We have very tiny hairs like this. This is a nail here, and you can see that this is quite very tiny. We only have these kind of herbs or subfruticent plants, and we had rosettes too, like this Brewaskia from the Tibet, also post-stray herbs like this Solanum dalibardiforme here. The epiphytic Solanaceae I reconstruct that we update the phylogeny from TINA from 2013. We include a little more species, a little more loci, 7 from chloroplast and 3 from nucleus, and just to trying to check in the literature for epiphytic cases of in Solanaceae. I know very well about the tribe Solandreae, that is an epiphytic group, but I discovered that there is other groups that are reported like epiphytic plants. This is the case in Lycianthes. This is here in the same clade, but in two different geographic places. This Lycianthes here is from New Guinea, and these two are from Central America, but they belong to the same clay. It's a small old world clay here, and it's a group of epiphytic in New Guinea, Philippines, in that area of Malaysia. Also, we had a couple of reported epiphytes in the African non-spiny Solanum. These two are from Madagascar. This is, I think, Solanum myrsinoides and Solanum trunsicola. We also had a few, this was a very nice surprise because we had a epiphytic potato here. This is two species, Solanum clarum and Solanum morelliforme. This is Solanum morelliforme that grow like epiphytes. They had this small potato here, and they used to grow in this place here. In Mexico, but recently they report, the specialists in this group report a second population in Bolivia, a very disjunct population. Okay, the distribution of Epiphytes solanaceae, I already mentioned that, but just to show you, we have this Lycianthes here in Malaysia, the Solanum africanus spine in Madagascar, the Solandreae here in green in the Neotropics, the population from Morelliforme from Bolivia, another from Mexico, and some Lycianthes in Central America. Okay, now I'm gonna talk a little bit about some vegetative adaptation in this Epiphytes solanaceae. This is a nice photo from Trianeana eca, I think this is from the Isao from Peru, from Cusco. And this is a very nice plant growing here like an epiphyte. Okay, just to mention that. I'm gonna talk a little more about these four traits: tuberosities, leaf texture and leaf arrangement, and adventitious roots. Because this is one of the, exploring the morphology in Solanaceae, I found that this is one of the characters you remember my introduction, could be one of the traits that could be related with the P59 in Solanaceae. We have here tuberosities. I have to say that tuberosities could be no homologous in all family, because we can have some stem tubers here, like this is a white potato, but we can have lignotubers. We can have these root tubers, like a tap roots here in this mandrake, in this genus Mandragora, or in this Jaborosa from the south in Argentina. We also have this leaf texture that could be changed from a membranaceous leaves to fleshy, coriaceous. This is a little fleshy too. We have cartaceous too. And the leaf arrangement, you know, the common arrangement in Solanaceae is alternate, but we can have some variation of that, like these fasciculate leaves in Lycium, or these cluster leaves in Quatresia anomala. I think this is going to transfer this to Lycianthes. And we have this imbricating Fabiana, and we have this common alternate in Chutesianthus. Also, I'm going to mention the adventitious roots that could be important for epiphytic plants, because the adventitious root, that this is roots that grow not from the primary roots of secondary roots, but grow directly from the stem or from the nodes. And this could be a good for mechanical support for epiphytes. Okay, and I'm going to map this in Solanaceae just to check what happened with these traits. I'm going to start with the tuberosities in Solanaceae. I know this is quite small, but we can see here we have it's absent in all these species, but it's present in 113 species, the species that I sampled here. And I just highlight here the 4 epiphytic clades just to check what happened with these clades. And we can see that occurs on tubers in in the Trisolandre, but this is not happening in Lycianthes, and it's not happening in the African spiny. And we had these two epiphytic potato that also had tubers here in the section petota. The tuberosities had evolved more than 20 times independently in Solanaceae. In some place like here, that I think this is Yosiame, Haborosa here, we have some scatterings across Solanum also, and maybe in Datura here that grow in the xerophytic places. Also, the adventitious roots is quite very common in the Trisolandre. This is a good indication of this trait. This could be related with the epiphytic life form. But this occurs also in some crepiers like near Embergia, or some, I think this is Petunia altiplana, I think this is the name, I don't remember. But this occurs also in some climbing Solanum here in scattered across Solanum. For example, I remember Solanum ovorbulifolium or Solanum ternatum that had adventitious roots too. And this is just an example from Solandra with the adventitious roots growing here. The leaf arrangement in Solanaceae is quite more diverse. We have different alternates that common here with more than 1,000 species, but we also have geminate, that is a variation or alternate where we have the leaves together, two leaves with the recouling set of these sympodial units. And we also have fasciculae leads in some groups, clustered and basal rosette. Cluster is the main leaf arrangement in the Trisolandre. You remember this kind of arrangement increase the exposure to light, and this is good indication of these traits related with the life form. Yeah, we also had this basal rosette here since some, I think this is mandrate and this stuff like we had the fasciculae here in Lycium, that is also a nice finding. Almost the fasciculae plants occurs in Lycium. We have this alternate and geminate in Solanum with some scattered imbricate. Imbricate is more in the species that grow very to the south, in the genera that grow very to the south, like Fabiana, this species that grow in Argentina. Okay. With the leaf texture, this is quite, it's a lot of variation in Solanaceae. Basically, Solanum had membranaceous and cartesious leaves. We have so many membranaceous in this part. The flesh is in the xerophytic Lycium and Nolana. That I think Michael Dillon and Vito Crepusco is going to talk more about this in the next talk. We had Cartesius and Coriaceous here. The main clade of Coriaceous leaves is in the Solandre, but we also have some scattered across the family. And just with that, I was thinking to map the unplanned interaction in Solanaceae, but we only had unplanned interaction in Solandre, and for that reason, I was not necessary to do that. I just put some photos here to show the interactions on ants. Usually lives in these plants. They can construct nests there too. We had true ant garden plant in Solandre, like this Marchea forbicarum that grow here and had these tubers that where some ants can live. Okay, and also I mapped some reproductive adaptation and the family, the pollination diversity in Solanaceae, just to trying to relate this specialization in epiphytic plants to vertebrate pollinators or to different pollinators like the trapline pollination, just to see that the Trisolandre has some is special in that way, and I record this. This states for the pollinators in Solanaceae. The commonest is bee. The Solanum is basically bee pollinated. But I found a high diversity of pollinators in the Trisolandre, but also in Nicotiana, and also in Euchromine. You can see the change in the colors here. In here, the colors are more or less constant. Here we had some moths. general insect in this set in this part, but along this clade is more or less constant. But we had some full of colors here, like a rainbow in this part, and also in Nicotiana and a little bit in Euchromine. And this is a nice finding too. Summarizing this, I can say that epiphytism has evolved at least 4 times in Solanaceae, with the Solandreae including the most of the epiphyte, more than 90% of the epiphyte occurs in this tribe. Tubulosities are present in 2 epiphytic clades, in the Trisolandreae, but also in the species of epiphytic potatoes. Adventitious roots are prevalent in the main epiphytic clade in Solandreae, but this not occurs in the other epiphytic groups. Cluster leads is strongly associated with the Trisolandreae, that is not occurring in the other groups. Lead texture is quite is a variable trait from Membranaceous to coriaceous, and is not a good, maybe not a good indicator of the association with the epiphytic life form. An association are exclusive to the main epiphytic clade. Pollinators diversity is high in the Solandreae, but the other epiphytic groups are pollinated by bees. This is like a kind of the summary of this first part. Now I'm gonna enter to the Trisolandreae, and I don't know what is going with the time. It's okay. And because, I'm going to focus in the Trisolandreae because this is the main epiphytic group in Solanaceae. I found that this group had the adaptation to the life form, like a clear adaptation to the epiphytic life form. And in this group, I was asking me these questions that I put here. What morphological groups can be recognized in the Trisolandreae based in molecular evidence? What taxonomy chain are required to reflect that results? How many times has epiphytic life form evolved within Trisolandreae? When I mapped across Solanaceae, I mapped like in 2 states, epiphytic, not epiphytic, but you remember we had different categories of plant that could be hemi-epiphytic, or could be nomadic pines, or could be facultative epiphytes. I want to go deep in this trait in Solandreae. One morphological trait could be associated with the epiphytic life form. We had a spoiler of that already, but I just want to see this I want to enter in the try and check that this is true or not. What pollination symptoms are involved in the Trisolandreae? We saw so many changes in the Trisolandreae, but also I propose to study better the pollination symptoms in the Trisolandreae, that we can apply the concept of pollination symptoms in the Trisolandreae. Okay, the first step was construct a phylogeny for the tribe. We went to the field, we went to the herbaria, and collect so many samples to extract DNA. We processed 60 species from the 80 that we know in this group. We can recognize 11 genera from this result. This is a phylogeny constructed with the best maximum likelihood tree using full plastomes with like a single partition. I also construct trees with the coding, with the no coding, gene by gene, with the base scheme partition, but all of this phylogeny was congruent in recover these 11 genera. For this reason, and this was the full plastome was the highly supported, and that's the reason because I choose this to circumscribe the genera that we recognize in Solandreae. This is 11 genera. We have the groups here, and we had a nice finding here, and we had 2 new genera, one at the like an early branch here in Solandreae. We have this Sarcophisa genus here, that is a resurrected genus. I'm going to talk a little more later about that. But we had 11. I can talk today. I can talk today from this 11 genera because it's going to be 2 hours more, maybe. I'm going to focus in the first 2 clades here, in the new genus 1 and in the Sarcophisa clade. But before that, I also because we recognize these 11 genera with morphological evidence, with molecular evidence, I also want to try to check what happened with this genera in the morphology. and I study these morphological traits just to trying to characterize every genus in Solandreae, and I found that most of these characters can be useful to describe, like a combination of characters can be useful to describe the variation at the genetic level that we had in the tribe. And this is good to can diagnosticate every genus in this tribe. Here, what taxonomic changes are required to reflect that results? Um, this is just a comparison of the genetic classification from the tribe Solandreae across the years. When it start with Myers, who described the tribe, Hunziker was in the 70s, in 80s until 90s in this group. He described a lot of the species. Also, Sandy was a lot in the 90s in this group, describing so many species. I work in my master thesis in this group too. And I put here some lines just to trying to show the changes. Was so many changes here when I published these results. Now we still have some changes, but there are not so many. But still, we have to transfer. We have to do a new combination, describe two new genera. We have a lot of many new species. It's quite interesting that we're gonna have nice taxonomy changes to accommodate all the species that we had in the tribe to these 11 genera. Mm, like as I mentioned before, I'm gonna focus in these two clades for now. And we're gonna continue working after my PhD in all these other with I hope with Sandy and with Tina and with other people that are interested in working in these groups. We have a new genus here that we're gonna call Docelia because this is the Spanish word for dosel, for canopy. Dosel is the Spanish word dosel that means canopy. And it's because these plants, these four species grow very high in the canopy. They are actually they are so hard to collect. We had three species described that we had to transfer from Archaea. We had one species that we are describing now. This is a genus that is distributed in Ecuador and mainly in Colombia. We had this across the Cordilleras in Ecuador and Colombia. This is the diagnosis. It's too long. I'm not worried about this. I'm gonna show some photos where I can explain better the character. But this is just to show that the description of this genera, of this new genus, or all the description, the diagnostication of this genera in Solandreae needs a quite long diagnosis because we had a lot of convergence in the morphology in these groups. This is the nice photos from this group. We have here the four species. We have Docelia epiphyta. This is gonna be this is the new species. This is Docelia willensis, and this is Docelia lopezi. That's gonna be the type of the genus. This is a nice, really showy flowers that you can see. This was the flower that was like a pendant in my first slide. And this is a photograph from Alistair Hay. And the main characteristic from this new genus is that they are always lianas that climb to the canopy, and after that just has some pendant branches like this that you can see here. They are not true epiphytes; they are hemi-epiphytic plants. But I'm gonna talk more about that later. We had these cluster leaves here. Always they have membranous leaves with a lot of simple hairs. And also we have these pointed fruits with two carpels and two locules. This is the main characteristics from this group. We also have this winged calyx here. Mm, okay. The second group that I want to talk today is the Gino Sarcophisa. This genus was synonymized by like for 200 years, but we resurrected this now. To include a species, we're gonna include Chultesianthus, that was a valid genus, Rajo guardiana, Juan Ulloa speciosa, that was described like a Sarcophisa, Solandra boliviana, Marquea grandiflora. This is currently we are circumscribing the genus to 20 species, 15 have names, and we had to describe 5 new species. This is distributed from Mexico to Bolivia, and this is basically growing cloud forest between 400 and 3,000 meters elevation. And the main characteristics of this genus is this is also a epiphytic group. This is a very diagnostic character for this genus. You can see here that all we have coriaceous leaves, and maybe it's not possible to see here, but you can see some dots here. And you can see this like a rutaceae or myrtaceae. You can see dots in this that is very strange in Solanaceae. But this is not this kind of glandular dot, but they are just glandular hairs, like at this type. This glandular hair that have this heat here, and they are sunken into the mesosphere of the leaf. And sometimes they are not sunken, but they also have this heat with many cells, and they are stipitate here. This is very characteristic from this genus. Sarcophisa speciosa had this kind of hairs too, and this is the reason, obviously because the molecular results, but the morphology is also completely makes sense. This is the variation in this group. It's quite a very heterogeneous group. We put all these species together based in the molecular evidence. We can see some morphology vegetative. They have some character like the hairs, but in the flower morphology they are crazy. These plants are just, we can have this globular inflorescence from 35 centimeters. We had this other globular inflorescence. We had this very tubular, narrow tubular flowers. We have funnel form. We had dark colors here, white. But we had this kind of very strange fruits here, baked fruits, that they are very strange. I think this is the first case reported of viviparous plants in Solanaceae. I don't know, maybe Sandy knows more about that, but I never seen viviparous plants in Solanaceae, but in the inflorescence of this species of, this is gonna be Sarcophisa globifera, have this small plant growing there. The fruits are very coriaceous, are very thick, the walls are very thick, and the scale is always open irregularly. In many species, not always, we also have this very splitted calyx here. And this is the variation in this group. We had to study this group better, maybe monograph this genus to understand all this variation. Okay, working in all this stuff, we discovered so many species, new species, but I just want to mention that to discover a species is not just go to the field one day and, oh, this is a new species, this is a new species. It's quite a hard work. You have to go to the herbaria, visit so many herbaria, local herbaria. You have to go to the field and collect so many plants, understand the variation of every species. And this, because this group was so poorly studied, we found a lot of new species. I'm gonna show you the species that I have live photos, and you see a lot of live photos from collaborators across South America. And this is just the, I think I put here 13 new species that we are describing now with Sandy and with Tina, and with other collaborators in Peru, in Colombia, and just species from all these different genera of the Trisolandra, like a Haucasiophyton. We have this, that is a new Docelia, this is a Trianea, this is a Juan Ulloa, this is another Docelia, this is a Merintopodium, we have Trianeas here, Sarcophisa, other two Sarcophisa, another Trianea here. Is a lot of new species to the pending to describe, because you know this epiphytic group was grow sometimes high in the canopy. They are quite have restricted distribution, and collaborated with a lot of researchers in Peru, mainly in Colombia. We start to, when the people here that I was working, we was working in this group, the people just start to send photos from different places, and we start to discover a lot of new species. But it's quite a hard work to understand all the variation to can say that this is a new species. Okay, now I'm gonna move to the another question that I had. How many times I have epiphytic life evolved within Trisolandreae? I think I'm running out of time, but okay, I'm gonna try to move. And you remember in the Solanaceae, at the Solanaceae level, at the family level, I showed that the Trisolandreae was an epiphytic, like a unit of epiphytic plant. When I start to check inside the tribe, I start to discover that these early branching groups are hemi-epiphytic. We also had a facultative epiphytes here that can grow on the story forest, can grow on the ground, but also can grow like an epiphyte. And we had true epiphytes here in this clay. And all epiphytes has evolved at least 3 times in this big clay here. That is the core Solandreae. But we also have some few cases in Sarcophisa. That is, you remember the highly diverse genus that we resurrect. Okay, when I start to check the tuberosities in Solandreae, they are not present in all clades. They are not in the hemi-epiphytic or in the facultative epiphytic, but they are only present in the holoepiphytic Solandreae and evolve at least 8 times independently. We have to check this better because we are not sure about the homology of these tuberosities, even inside the trisolandreae. What morphological traits on the Myrmeco, I don't know how to pronounce it, the associations is also present only in the holoepiphytes in the trisolandreae, but just in a few groups and evolve at least 3 times. You can see here some of the evaluation that I will show. Okay, this just to I spoiled this before, but obviously these traits are related with the epiphytic life form, and not just with the epiphytic life form like a general concept, but with the holoepiphytic, with the plant that are obligated to live like an epiphyte, that this plant never had contact with the ground, and these traits are very associated with the holoepiphytes. That is quite a nice finding. And we had this flower morphology that is quite crazy for this small group, and we want to just trying to figure out what happened with these plants, why they are so diverse. We had a species with 5 millimeters here, and we had a species with 40 centimeters here, and we want to try to understand if this large flora diversity are associated with pollination syndromes in this group, with the increase in function and the use of functional groups of pollinators in this tribe. And for that, we had to go to the field because we have just a few records of visitors of pollinators in the trisolandreae. With the support of the Davis Expedition Fund from the University of Edinburgh and the Royal Botanic Garden of Edinburgh, we go to the field with, I have to say thank you to Juan David Tovar that helped me a lot with the field work. We visit Colombia and Panama. Using this camera, when we had this species growing close to the ground, we put the camera here in a tripod, but when this plant was growing high in the canopy, we had to attach the camera to the trunks here. Also recording at night because we had the option, we had maybe the possibility of some bad visitors. I had to record at night too. With this field work, we start to record pollinators in the trisolandreae, had more records to kind of understand better the pollination in this group. And we had these hummingbirds here visiting Sarcophisas. This is another Sarcophisa, other Sarcophisa. This is all Sarcophisa here. All these species are hummingbird pollinated, at least visited by hummingbird. Also, we had these bad pollinated plants in the trisolandreae. Most of these photos was we record at night. One that we record some bad visiting Trianea nobilis here in Nariño in Colombia, but most of these photos was stolen. They are from Nathan Mushala that was working for a long time in this group. I had some conversation with him a long time ago to talk about this, and we shared these photos. He shared these photos with me. And also, I started this photograph from Disochroma from a paper by Sassima, and also some photo from Melintopodium that someone, Merlin Turtle, put in Google, and I just was hunting this kind of photographs in internet, just trying to have more information from these pollinators. This comes from the fieldwork from Juan David. We had these bee pollinated flowers. They are small, tiny flowers, green, mainly campanulate flowers with cone-even anthers here that they form a cone. We had a different kind of Alitide bees. We had Euglosine bees. This is another Alitide bees. I have to say thank you to a lot of people that helped me to identify these bees, to Victor, to Antonio Aguiar, to Edith Trujillo, that helped me a lot to identify these bees, because, you know, working in Solanaceae is so hard to try to identify this. Also, we had bees visiting these big flowers. We can know that these flowers are quite similar, but here we had a high diverse morphology with different colors, with different lot size. We record a lot of bees visiting these. These are mainly Euglosine bees visiting these flowers. Also, we had some other visitor like a moth. You have to believe me that is a moth here and is another here. We also had some scarabate beetles here. We had a small tiny Tawani fly here, and this moth, at least they are reported like we are reporting this like a pollinator of this Sarcophisa species. Mm, Along with the fieldwork, we went to the herbaria. We measured several morphological traits that we read in the literature. We relate this, following also the words of Laura Lagomarsino. I have to say thank you to Laura for all the support with this part of my thesis. We measured all these traits in the species of Solandreae, to trying to relate the morphology of these flowers with the record that we had from the field. When I put this morphology in a principal component analysis, trying to put also the records that we had, they are kind of, I can see here, but they had some circumference in black here. They are the confirmed records from the field. And we start to see that all these morphology are distributed in the morphospace in different parts. At least the hummingbird pollinated flowers are in this part. We have a lot of bud pollinated here, and we had these small bee pollinated green flowers with campanulae and condiment anthers here. But we had this mixture here. We had crazy morphology, flower morphology here, and we had also mixed records here. We had bees, but we also had moths, but we also had hummingbirds. And we think that this species in this part of the morphospace could be had a mixed pollination syndrome, a mixing insect with vertebrate. Um, and when I put this in a phylogenetic principal coordinate analysis, I can know that some species that are hummingbird pollinated, for example, they not belong to the same clade. For example, this is a Juan Ulloa, but this is a Haucasiophyto that come from here, and a Sarcophisa that come from here, and we had another, I think this is a should the cyan to the now Sarcophisa. We can see that this exists convergence to the at least to the same morphology, and we are associating this convergence to the same morphology with the use of the same functional group of pollinators. This thing happened here with the bee pollinated. We had Haucasiophyton here, but we also had some Meritopodium. We had some, I think they are mainly Haucasiophyton, but we also have Ectosoma here. But pollinated flowers are basically are from different groups too, like Atrianea, Meritopodium. And the mixed group had everything here. This when I classify this according to we are predicting the pollinators with the record that we had and with the morphology, assuming that this species had a confirmed record from the field, and this is this part on the morphospace, and we had one species that we don't have records but had the same morphology as in the same part of the morphospace. We are assuming that this species could be pollinated by the same functional group. But we want to test that. We do the linear discriminant analysis trying to discriminate these pollination syndromes, at least for the bee, the small green condiment anthers flowers. We had that is completely a separated syndrome. Hummingbird is also separated, but mixing a little bit with the bud syndrome, and we had this mixed syndrome here in the middle of bats and hummingbirds here. Summarizing this, possibly we have 4 syndromes: hummingbird, bat, bee, and a mixed syndrome. We had to test that in the field. We had to work more in this. All epiphytes tend to have specialized syndromes. You can see the mixed syndrome placed in this part, but in the all epiphyte we have more like pollinated by hummingbirds or by bats or by bees. They are tend to have this specialization. And our morphometry study, like a conclusion, combining fieldwork with the flower morphology indicate that the pollinator syndrome could be a useful concept in Solandreae and likely explain a large part of the floral diversity that we observe in this group. And just to mention here, I highlight here the new species that we discovered in this study, and just to show that we need to study the material in the barrier. We need to go to the field because it was a huge gap. Without this species, it's gonna be the same results. Without we go to the field, we study this specimen in the barrier, in many local herbaria in South America. This one, the result gonna be other. And just to leave that like a message. Thank you to all people that support this work. Was a lot of people. I don't have the space here to mention all of them, but obviously I had to say thank you to Tina Sarkinen, my supervisor in Edinburgh, and Richard Mine and Catherine Kinder for all their support. To Laura to help me a lot with to understand the pollination syndromes, the methodology, the R scripts. Also to Juan David for the help with the fieldwork, and all the Solanaceae, Solanologists community, and Stephen Dosworth for helping me a lot with the molecular data, Peter Potsai to understand the plastome in Solandreae, and Gaurav to help me with the bioinformatics from this. Some problems with the bioinformatics sometimes. Okay, and all my sponsor, the Davis Expedition Fund, the Royal Botanic Garden Edinburgh, University Edinburgh, the Fundación Ceiba from Colombia that supporting me with a grant to go to Edinburgh to study my PhD, the Jardín Botánico de Bogotá that always support me here, and I always had a herbarium where war and with access to everything, and it's always it's always nice to return to the Jardín Botánico of Bogotá and to the Systematics Association for the economic support too. And this is all. This is extra. Thank you for your attention. And someone had question. And sorry, it was so many information. Was a lot of information. Not sure that you had question.
Thank you very much, Andres. That was amazing. There are so many comments. So, with congratulations, of course, because this was very, very beautiful slides. Yes, it's a great, an amazing amount of work, and yes, it's a very beautiful group as well. So, well, there's a lot of comments just saying congratulations, wonderful plans and presentation, and all the pictures. Everything is amazing. And Sandy Knapp, she was saying something about the anthers. I don't know if you want to turn on your microphone, Sandy, to that comment.
Sure, Andres, you know the ones that have the kind of anthers that are connivant into a cone? Mm-hmm. Do the bees buzz those, or do they just glean pollen? How do they interact with that cone of anthers?
Oh, these anthers are quite similar to the buzz pollinated anthers because they form this cone, and they open by longitudinal slits, but these slits open at the very apex of the anther, like simulating a polycidal anther. But I saw this a couple of times in the field, and I never seen the bee buzz in the flower. But it's something that we have to study better. Maybe.
Yeah, that's really interesting. Thanks.
Okay,
Sandy, then we have another question of Richard Olmsted. Do you want to turn on your microphone and do the question?
Sure.
Oh, you're muted. Okay, now you're.
Tina tried to answer this in her chat too. So, but I'll ask it again. In your mixed pollinator group, is that a mix of species that have many different individual pollinators, or is it a mix of species that each has multiple pollinators? If you know what I mean. Like, is there one species that's bird pollinated, another species that's bat pollinated, another species that's moth pollinated? Or does each one have many pollinators?
I think it's the second case. We have many pollinators by each species. We had records for at least 2 that are visited by moths, by hummingbirds, and by bees, the same species.
Wow.
At least we had this from the field. But yeah, this group did a lot of the study. You know, we need to go to the field to study the species from a strange morphology, a strange color, like a very dark colors with different lobe size, with a huge corolla size, like the Solandreae. Yeah, that group need a lot of work to can say that this is a mixed pollination syndrome. We only had this indication that could be the case, but yeah, we need to study that a lot more.
Thank you.
Thank you.
Thank you, Dick and Andres. Then there is a question from Ellen Dean. Ellen, do you want to turn on your microphone? I can do it for you.
I just had a question that if there was any evidence of a switch from nocturnal moth pollination to bat pollination within this group.
The moth record, actually we only had 1 record of moths in this group. This Sarcophisa. That is I think it's in, let me show this here. I think it's in this part. And all these came belong to this mates group. Yeah, with the records for moths and change from moths to bats is no, we don't have that kind of information yet. Yeah, we had some bat pollinated flowers here, but the mod is including the mates for now.
Okay, thank you.
Thank you.
Thank you, Ellen. And there is a comment that I would like to highlight from Mark Hughes, that he said that it's a great presentation and good to see phylogenomics being combined with lots of careful field and morphological observation. And this is actually the really great of your thesis and your work, that you have combined all these new methodologies with all the taxonomy and the monographic work. This is amazing. So I want to highlight that. And then there is a question of Gustavo. I don't know if Gustavo, do you want to turn on your microphone?
Hello, can you hear me?
Yes, perfectly.
Yes,
cool.
Gustavo.
Yeah, I was not sure that it was working, my mic. So yeah, first, congratulations, Andres. That's a very nice job. Yeah, you study a lot of features there, so I'm really impressed. I am just curious about the seed morphology and dispersion. I don't know if it should be relevant. I will expect that in epiphytic plants, the seeds should be small to just don't drop to the soil, and of course the dispersion should be for something that is high in the canopy, maybe the birds or the wind. So how much do you know, or how much is known about these features in the epiphytic Solanaceae?
Thank you, Gustavo, for this question, for the work. We only have a very fragmentary information about that. I know that we have some information about eliosomes in some seeds from Markia, that this is these ant plants, the Markia formicarum, and this species that grow in this nest of the ants had reported had seed with eliosomes. That is an adaptation also to be dispersed. But this is the only information that we have. Yeah, this is another topic that we need to study more. The seed morphology in these epiphytic groups.
Okay, cool. Thank you.
Thank you, Gustavo. Then there was the question of Edeline Gagnon, and she already answered herself the question. So I'm going to continue with another question from Laura Lagomarcino. And do you want to turn on your microphone and do the question, or should I
Yeah,
read
sure.
it?
I can do that. Hey, Andres. Hey, Rocio.
Hi, Laura.
Congratulations. It's so great to see how far you've progressed in the last couple of years. This is a beautiful talk. And I just wondered, and I thought it was really interesting that, Solandreae has some of the only times in which ant-plant interactions seem to evolve within Solanaceae. Do you see any interaction between ant interactions and floral traits, pollination syndrome, size, or nectar production, or anything like that? Or is it more elevation, maybe like an elevational thing?
Sorry, Laura, can you repeat me that? I had
Sorry,
problem with
yeah.
I have problem with my volume.
No worries. Do you see a correlation between when those ant-plant interactions evolved and pollination syndrome, or other aspects of floral biology like nectar volume or anything like that?
You say these traits are related to the pollination, like when we had bud, we had increase in the nectarifer and in the size of the nectarifer's days, for example. We had yeah, I know. I'm not sure that I understand the question.
Is it
Sorry, is the volume probably that it's a bit low? So I can read the question otherwise. Yeah, because it's if there is any correlation between the ant interaction with
The ants. Okay. I'm sorry, I missed that part. Sorry.
or with the altitude, or yes, you have seen any correlation with any flower trait, flower related trait with the ant interaction?
The ants.
Yes.
Yeah, the species that are associated with ants are always growing to low elevation, like the Amazonian basin, but they never interact with flowers. They used to walk in the flowers, but at least I never seen that the ants, for example, walking in the into the anthers or in the stigma. I never seen that. They just it's kind of patrol, just walking around the plant. I think just protecting maybe the plant. It's some information like I mentioned before with the dispersion in some markia, in some true and garden plants, but no more than that. No, the ants are more like these ants construct nests in the branches, sometimes in the inflorescence. Like they construct nests there, but I think it's just because the mechanical support of the peduncle, something like that.
Awesome. Thank you, Andres.
Thank you, Laura.
Thank you, Laura. Yeah, that's great. And then there is a question from, well, a lot of congratulations, of course. And Gabriela Doria has a question. Do you want to turn on your microphone?
Well, I guess it's similar to Ellen's questions. I was just curious about the moths that you didn't mention, but yeah, you.
Oh, yes,
So
right.
I guess it's more still missing information. Are there any flowers that you would expect to have moth pollinators, but you haven't seen them yet?
Yeah, I had the feeling that the all Solandra are moth pollinated. These big flowers, the huge Solandra flowers, and actually I can see here Laura Lagomarcino with a flower of I think it's Osa or Cubanicola is the other Cubanicola, the other genus of Rubiaceae, that had these huge flower pendant flowers that had a morphology very similar to this Solandra. And we put the cameras in Panama in Solandra brachycalids. Maybe Juan David can tell more about that, but we never seen a pollinator at the day or at night. I don't know what happened with Solandra, but the flower, they had a very narrow tooth, very long, like 20, 25 centimeters. It's not for hummingbirds, it's not for bats. I guess that it could be that group, this huge flower could be moth pollinated or pollinated by moths and another thing, but we don't have information from the field. It's quite strange because Solandra is a cultivated group. They are not too common, but you can find this species in the field, and No, we never seen a pollinator. And searching in Google is nothing about Solandra, but I guess that could be more pollinated. That could be more
Thank you, Andres.
Thank you, Gabriela. And yes, about that you were mentioning, there is this discussion that probably Juan David Tobar can say something that he is saying about what about the mouse that you found together, like in Sarcophisa. And actually, Leandro was also asking about that, and Tina was also responding. So I think probably Juan can say something. If he's still here, I'm not entirely sure.
Juan David?
Okay, so probably, or his microphone is not working, but so what can you say, Andres, about that mouse that you found in visiting Sarcophisa?
The mouse, the rodent, yeah. I think that was the rodent was just walking around, and this flower had a very strong smell, and I think this rodent was attracted by that smell. But I think it was I checked the video many times, and the rodent was just walking. This is a kind of cup, like a bit surface of this flower there, and they had a strong smell from the middle. And I guess this rodent was just walking around and just was attracted for that smell. But I think not sure. We never can say discard something, but at least with the information and with the video, I can say that that rodent was just a occasional visitor.
And then Leandro probably is here. Oh, yeah. Go ahead.
Yeah. So just to complete your observations, we have seen that the flowers of Dispersion atlanticum, they grow really low in the canopy. They actually, some flowers emerge 30 centimeters from the soil. And it was really intriguing to us what could be the pollination of that, because it's really low, it's really close to the soil, you know. And
yeah, I know. Sorry.
I stood observing for a couple of days when I went to the field to collect it, but I couldn't figure out exactly what was the nature of the mouses visiting it. But there are some mouses involved, you know, but I can't really say that they're pollinating. But it's interesting to look for that too. Maybe we can set up a field trip for that.
Yeah, that could be nice. And we discovered a new species in Peru. This is one of the Haucasiophyton, has the small flowers. This species, this flower, that plant produces the flowers inside the soil. It's like the flowers are very close to the ground. It's, growing, just producing flowers at the ground, just in the middle of adventitious roots. That plant produced flowers there, and I was just wondering what happened with that flower, and I was thinking in rodents too, but
right.
Yeah.
And it's an interesting development feature too, because like for Dispersion atlanticum, for example, it's a very high epiphyte, so the leaves are in the canopy, you know, the leaves are dozens of meters away, but the flowers are all close to the ground, you know, they are in the trunk close to the ground.
Nice, so nice. Yeah, we have to study that more.
That's great. Thank you, Leandro. Then there is another question from Richard Olmsted.
Andres, that was a fabulous talk, I should say. It was so impressive. I noticed in one of your slides where you compared the classifications of Solandreae, that in your unpublished work, you listed 2 new genera, but you only told us about how's it called? Doceras?
Docelia.
Oh, you want him to reveal all of it, yeah? I'm teasing.
Is there another genus coming from this?
Dun, dun,
yeah,
dun.
we're gonna call that then Carmen Emilia by Carmen Benitez in honor to her. And this is a genus of 3 species. This is the Facultative Epiphyte. This species grow in the Choco forest, and they used to grow in the ground like a red shrub with this kind of umbrella, with these clustered leaves growing like an umbrella. And this produce a new growth from the middle of that clustered leaves and produce a new umbrella and grow in that way, like like the Croatresia anomala. These are very interesting genus too. But we don't have time today to talk about that because I was extending too much with the time. Sorry. But we can talk later in another
Thank you.
opportunity.
You need to present a full conference, Andres. So many things to talk about. And there is another question from Stacy. Stacy, do you want to turn on your microphone? Or do you want me to read?
I think Stacey had to go, but she'd like to hear the answer so that she can watch the recorded.
Yes, of course.
Yeah.
She said, I would like to know if at the family level, if there are an apparent reversals from epiphytes. Maybe Andres said and I miss it?
We had this, we was talking about that. This new genus could be a reversal because this is in the middle of, this is at the base of the holoepiphytic group. And they I can show the what is that? Let me remember. Not sure that I had this here. I think it's here. It's here. We had this epiphytic here. We had this switch shift here to the holoepiphytic that we had this facultative epiphyte here in the middle. And I was thinking that could be a reversal to the terrestrial life form, but not sure about this. This species most of the time grow on the ground, but I also found this in the main part of the tree, like in the trunk, in the main trunk, growing very close to the ground, like in the first 1 or 2 meters from the trunk. They can grow like an epiphyte in that part of the tree. We have some kind of stratification with this epiphyte, no? And this case of facultative epiphytes sometimes grow very at the base of the tree or grow on the ground.
So in some sense, here's I guess continuing the question. Do you think it's gradual evolution from terrestrial to hemiepiphytic to facultative to holoepiphytes, or do you think it's a reversal?
I think it's at least from hemiepiphytic to holoepiphytic is like to go from terrestrial to holoepiphytic. The hemiepiphytic is like a transitional step. At least how I can see this, but the facultative could be also part of this transition, or could be a reversal. I'm not sure about that, Tina.
Or
No, I don't know. What do you think?
Because it's facultative, it's a bit more labeled, the epiphytes. So it's more reversal in some way because it's still facultative. So, I don't know.
Yeah, I record this like a reversal in my thesis. But yeah, because this is the most common is the commonest is the terrestrial.
That's great. Thank you. And then we have, sorry, I'm a bit lost. After Stacy, yes, Clarines. She has a question. I don't know if she's here. I can read it. So yes, she said that perhaps she lost some information. And finally, the big polytomy that you found in your master thesis, was it resolved with the plastomes?
That's a good question because yeah, we had this result with the plastome that we improved the result from my publication from 2017, but we still had to work especially in this core Solandreae. We still had some medium to sometimes low support here, and I'm gonna keep working in this. Actually I had some sequence from this 353 library from Pactol to process, and yeah, I had the data, and I wanna try to explore more that this part of the tree, because we still have things to do in that part. Yeah, we improve, but we need to work more in that. For sure.
Thank you. And then Federico Roda has a question.
Federico.
think he is still here, or he already left.
I think Federico says he's left, yes.
Oh, okay. Okay, sorry. Yes. But then probably, yes, he can email you the question. And I think, let's see. So there is some comments, but that was so last questions. I don't know if I missed someone, please, because there were so many questions. It was so interesting. Please let me know and turn on your microphone and let me know that if you want to ask an additional question. Otherwise, we are finishing the seminar. So thank you very much again, Andrés. That was amazing, really amazing talk. And I'm going to say, well, first of all, I'm forgetting Happy New Chinese Year for all the folks from China and other countries as well. And next Friday, we have an amazing joint talk between Michael Dillon and Victor Kipuscoa about Nolana, and they're going to talk. Their title is Solanaceae 6 largest genus with hyperdiversity and within hyperaridity. So it's going to be about Nolana, of course. And so we wait. Well, we welcome you, all of you, like next Friday to continue with these Solanaceae seminars online. Thank you, everyone.
Thank you to everyone
Thank
for the
you,
attention.
Rocio, for